record. So as I mentioned, Piper and Sean are away on vacation. Speaking of Piper, I asked Piper and Caitlin to reorganize the AI-driven Anthrobot manuscript draft. They circulated it, but I want to make sure that Caitlin's part in that manuscript is better integrated into the flow of the story. So Piper and Caitlin and Gazem are in the process of reorganizing it at a high level. And then for the folks that were invited to go in and supply edits, perhaps you can do so after they've done some high level reorganization. I know Gazem has graduated and Piper is not going to graduate for a while, so there's no real time pressure on that particular manuscript, I think. Obviously, earlier rather than later is always good, but I'd rather get the story straight before we send it off for review. Everyone in agreement with that who's on that paper? Silence implies assent. Okay. All right, I'm happy to open the floor for updates. And then if we finish updates before the hour's up, I was hoping we could just review some of the biosense stuff, system integration between AI and the biology side of things if we have time. Okay, updates, please.
Not an update, but can I ask, I know Piper's on vacation. I'm not sure when she's back. It's fine if it's just next week, but if she's on vacation for a while, who can I get some either swarm or just bot trajectory data from? I just want to run it through the current model I have to see how accurate it is on like not manufactured data on actual data.
Can you, Amanda, can you say a little bit more about what you want to do? That might help.
Oh, yeah. I just need either a video or just the like XY coordinates of the Xenobot over time. I'm going to be sending it through a deep neural network that will try and build an equation, a differential equation that describes the movement of the Xenobot over time. And from that, I can from the equations that model produces, I can get the predicted trajectory. I just want to see how well this model is doing on actual data.
So do you need new videos that beyond what you guys already have for the analyses, or you can use the same ones?
I can use the same ones. I just need to know who has them, so I can pull them.
I would say Caitlin, Amanda.
Okay.
So she's got a lot of the raw Xenobot data, and also all the imaging software to pull out the trajectories if you need it. I don't think we need anything fresh from the Tufts folks yet.
Yep. Okay. Perfect. I will.
By the way, if just in case, if you want to, you also got some Anthrobot data that Nick Davey sent recently. That was the Anthrobots that were exposed to the different disease, the patient's matrix. If you want to, it's not the same thing as the Xenobots, but if you want to test out your method, go for it.
Awesome. Thank you.
You guys already have all that.
Will do. Thanks.
I know Thomas was starting to put together some cloud-based folders of exactly that, just videos that have already been processed and published that we can just use as a proving ground for some of our stuff. So you might,
Okay,
Andy, you might also want to reach out to Thomas.
Yeah, I'm here. I'm on.
Oh, there he is. Okay. Yeah. Great.
All right. Awesome. Will do. Thank you.
Other updates?
Yeah, I can present something that I recently published.
Not published yet.
Not submitted to a journal for peer review. Yeah. Okay, can you all see the screen? Okay, so typically what we have is when humans want to model a dynamical systems, they observe the system, say in this case, a single pendulum moving around. And then a human chooses state variables that are suitable to describe the dynamics of systems. In this case, they could choose the angular position, the angular velocity, and then they would have some way of formulating a dynamics model. So they could use Newton's laws of motion and then get this ODE to describe the system in terms of those states. So they could also use alternative state variables, which are energy variables. So this could be potential energy and kinetic energy. So this is expressed as a function of these positions and angular velocities, but then you could have different formulations using different variables. And we can do this for simple systems. So the question is, for a complex system, it's not clear what the state variables would be. So the next step was, why don't we feed in these observations to a neural network, and it also finds some set of state variables. We call them neural state variables. And then we decode it, but instead of predicting the states, it predicts what happens to those frames in the video. So the question is, given that we can do this, we can find neural state variables that the neural network compresses down to, these set of observations that even a human can find. Is there any correspondence between neural state variables and human state variables? Like, is there something that we can learn from what a neural network compresses down to? So the one way to address this is, like, I mean, the neural state variables, they have different units. They don't have any specific motivation to capture, say, position or angular velocity. We don't really know, because they're trying to reconstruct frames. So how do we even compare these two? So we thought of interpreting them using information flows. So one way to do this is to ask, what does this variable 1 tell about variable 2? So what does angular position tell about angular velocity? So by comparing the information flows, you could sort of associate that they're capturing something similar to these pairs and whatnot. So we did that, and with the help of Thomas's guidance on what kind of information flows we could compare it to, we found that that it captures the distribution. There is a distribution of information flows that it is closest to. So in this case, it's capturing something like the second derivative of kinetic energy and the third derivative of kinetic energy. We tested various different combinations of human state variables, and this is the distribution that we find. So this is the result that we found for a simpler system like a single pendulum. So the question is, how can we use this method of analyzing or interpreting these neural state variables to a complex system? So that's where I'm beginning to use this pipeline to apply to a set of Xenobits videos. So in this Xenobit video, we could have positions that we could use to capture cells. These are human states that we can describe. You could have relative distances between these. You could have morphological features. So we don't really know what's predictive in these systems, and we don't really know a way. So the best way to do that is to use a neural network to capture the relationships in the video, and then find information flows that are closest to these pairs of or human state variables, and that could inform something about the system. So that's sort of like the overall like outlook of using this pipeline into discovering state variables in a complex system. What do you guys think?
So one thing that I think we should note, Krishna.
You're cutting off.
We can't hear you. We heard you for a second, then you went away.
Testing, testing.
Oh, you're back.
Okay. So one thing that we should say is that Krishna, you're not directly comparing the mutual information between state variables and neural and human state variables, right? Because you're doing a more there's an additional step in there where you're computing the structure of each one independently and then looking at the distance between them, right? I just want to make sure
Yes.
that that's clear.
Yes, yeah, that's the further detail of this, is that we are actually comparing what this variable tells about this variable, not across these two different systems. So we are measuring the distance between how this variable n1 tells about n2 across these different frames, and then comparing it to what each of the angular position and angular velocity tell. So if they're capturing something about angular, so we are just comparing information flows within this system and then measuring the distance between them. Yeah.
So-
Krishna, sorry, can you go back to that table that you had with the different colors?
Yeah.
Can you explain that again at the bottom, that the first and second derivative are what most closely related to the human state variables? Do I have that right?
Yes.
And how did was this training on a single video or with a set of videos? Like, how was this data generated? I'm just trying
So
to
this-
understand what's going
so-
into the system.
Right. So this data, so let's say we have 60 frames in a video, and we have 120 such videos, and we use 120 such videos to compare to get one single number of mutual information, and then we conduct several multiple trials of these videos on different data splits where we get neural networks, and then we associate the distance between those two mutual informations.
Got it.
And we also have a null model of mutual information. In this case, we compare. So for example, you could have position and say pixel or say pixel of the intensity of the pixel at position x comma y. They are not correlated. So we could have meaningless, like the mutual information of those two variables could be low. So we shouldn't use that for comparison. So to weigh that, so I normalize the mutual information with the null model where I shuffle one of those states. So the mutual information should be really low. They're uncorrelated.
So just to put this in a little bit more context, this is part of the ongoing effort to try and explain like what's going on in terms of self-assembly without us having to presuppose, the state variables or the descriptors of what's going on.
I mean, it occurs to me that like this is super cool, and it occurs to me we could use this method for all kinds of, like, for example, for our gene regulatory network data, right? That just the whole idea of not having to know ahead of time what the relevant observables are is awesome.
So I assume this is set up. Actually, I shouldn't assume. My question is this sort of set up like a principal component analysis where is it difficult to pull out from this first and second order derivative, like what the AI is actually zeroing in on? Can we ask it that question, or is it sort of very abstract how it's doing this math?
So the question is, when you train this AI, is it hard to recover
Yes.
whether NSVs are really pointing to some derivative of, say, position or velocity, right?
Correct.
How do you do that? So the original authors of this paper, in fact, trained a nonlinear regression model to predict the next states of position and angular velocity, and it does well compared to, say, higher dimensional versions of these latent vectors. So if you have a latent vector that is, let's say, 100 of these, and those 100 vectors predict angular position and angular velocity poorly than these two intrinsic dimensional neural state variables. But they don't, just using them and finding a function to predict, say, a particular angular position, angular velocity doesn't say what they really track, right? So it's not easy. There is no direct correspondence of finding a relationship in exactly what they track. So this is why we need to compare information flows and figure out that they don't have intrinsic motivation to track position and velocity. So the only way to do that is to have a loss function or constrain the latent embedding, but which we don't do here. We just reconstruct the mean squared error in the loss, like in the pixel reconstruction error. That doesn't motivate the latent dimension to track a particular derivative of position or velocity.
I get so sorry, just I want to make sure I understand this because this is really cool. All that is essentially all that your algorithm is seeing is for each cell, it has a position, and each cell has a positional velocity based on each frame. And that's all that it's not doing, doesn't understand like how many other cells are around, anything about the environment, any other features other than sort of a single positional velocity. Is that correct?
I mean, it sees the frame of the pendulum here.
Yes, I'm thinking when we sorry, let's extract to the Xena bits. So imagine you give it a file there.
Right.
Is it just looking at each individual cell or little blob, what its positional value is, or is it also taking into account like what's nearby, what was nearby? I'm just trying to understand from the point of view of the algorithm, what is it extracting from those frames that you're showing me that are animated?
So note that I have not shown this plot for the Xenobits video. This is for the single pendulum.
Yep, no, I get that. Yep.
So applying this would actually reveal what they actually track. I mean, we should propose that are they looking at blobs? Are they looking at, say, shapes? Are they looking at relationships of related distances? We should propose plausible variables that matter to the dynamics. And then we could ask the question, okay, by reconstructing the frames, what are they actually close to? What are they using? The information flow that are closest to. So that analysis could inform us that what's useful for predicting. Yeah.
Yes, I'm just so the only reason this matters is I'm thinking about this only from the point of view of the biologist, right? One way is we give it the variables we want it to track. The other way is we say, here's the frame, extract whatever you can extract and make a principal component analysis using number of cells per frame, distance between cells, blah, 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 and it comes up with something. The only problem is if we don't know exactly what's happening, you should just make sure we know that because, right, like in the human model, when I'm looking, it's pretty easy for me to identify if we've changed something experimentally, right? Like maybe Pi's videos are at a slightly different speed than mine, or the magnification is different, or the cell size is different. If the AI can't know that, we may, if we get a failure state where the AI is failing to predict something, we may not know if it's because the model's wrong or we've changed something about the data going into the system. And you might not know that either. So what I don't want to do is give you data and it keeps failing. And then in 3 months, it turns out like, oh, I captured this on a different microscope and the colors are different and that's freaking out the AI, right? Like, I don't want you to be digging into the algorithm and just making sure we're communicating in the data acquisition process to make sure we're not accidentally creating problems for you.
Yeah, so that process of like pre-processing, what should the AI model train on? Like, for example, like if you shot a blurry video and then it fails, of course, the data that you feed in has resulted in that failure. So you could make that. But for other failure modes of, say, a different dish or different sampling frequency, that is yet to be determined. I mean, sampling frequency is another thing that might affect the system. So.
Another way of looking at this is I think when we apply this to the xenobits or any other biologicals, like the best case scenario is with enough data, the AI model gets really good at prediction, and whatever the info flows are among the neural state variables is actually far from what we would pick as, usual suspects of state variables. So the AI says, I see, basically would be saying, I see something that you don't. So I think it could be helpful to actually rule out some of the obvious state variables. I don't know, I'm more fuzzy on how it would lead us to, unknown unknowns, but that we can work on that. But at least I think we could make a dent in it's not this, it's not that, or it's not just this, it's not just that.
Krishna, the other thing that's unrelated that I was going to ask about something like this is how scalable, like as far as number of cells, would something theoretically something like this, like you've cut this video down to something that's smaller than the original video, right? The animated
Right.
GIF here. Like, do these things get bogged down? Like, is it an exponential amount of compute as the frame gets bigger, or is that not really how it works?
Yes, the input size, in fact, makes it hard. Like most of the video prediction models work on like 64 by 64. Right now I'm using 128 by 128. And of course, most of the models, they're like billions of parameters. I use like a million parameter model. With more parameters, you're going to get a better, more ways to find the reconstruction approximation. But as you increase the input video size, the frame size, it's going to be exponentially more increase in compute.
Got it. I mean, something that's just, it's also something interesting to me that's a little bit unrelated to this is you could imagine giving, let's say you, right, I understand you're using sort of a toy model now with artificial data with the pendulum, but imagine the videos that we've supplied you. A cool question might be like, if we give you the full frame video and you run the full frame versus we chunk out the video into 64 smaller GIFs and you run each of those separately, is the answer the same or different, right? By having the full frame, is there more information gained, or is it sufficient to just run small windows of data? And I don't know biologically, like, right? I also am imaging a subset of the whole dish. Maybe if I imaged a bigger region of the dish, there would be more information, or maybe not. But that's the kind of thing that's also useful for us experimentally to try to understand, like, what is the right scope of data collection that would be best for these types of models. And so we can even think about for MomBot, like how big or small of an area is sufficient to understand how to get the cells to move around. And that does impact some of how we're designing the system on the optics end as well. So just something to think about, like not something for you to solve, but like an idea as you're developing these things, like what kind of resolution do we need? What kind of capture rate? How big of a field of view is sufficient?
Good.
Krishna, didn't you kind of do something like that? I feel like I remember a conference submission a few months ago where you were comparing predictions on the whole video versus you'd subsampled the video and aggregated and then sort of re-aggregated it. We were looking like a whole, like, is there information in the whole that's not in all of the local parts kind of thing?
Yes, in that case, both of them are of the same input size. So if you have a bigger video, I use image resizing. So it's basically a blurrier version of the whole video. It's not the original input size. Like, let's say if you have a 1080 by 1080, I can basically subsample it to 128 by 128. So you have all those pixels, but then they're like an average of those 128 by 128. Instead, if you take chunks of those multiple chunks of 128 by 128, they are the original resolution. So it won't be a good comparison. That's what I thought. Like when I did that, the result that I found was there is no difference in predictive accuracy. There's no significant difference in predictive accuracy if you use a data set that uses the whole video, which is subsampled into the same input size, or if you use chunks of those different videos of the original video.
Okay.
But that was also for, again, maybe not a sufficiently sized data set. So I think we can revisit all of this, as MomBot gets built and try and do this at larger scale and ask the same questions.
Yep. The only thing is for you guys, right? Like I can design the system, but what will be harder is a year from now, if you're like, oh, I really wish I had 3 times the resolution. Oh, I really wish I had 4 times the optic space. Like as we zero down on components, those changes will become harder. And as I generate biological data, I'll give you what we see in the lab from the MomBot prototype. But just keep in mind that if you see something where you're like, oh, that intuitively that's not looking great, or like the image is really blurry, I'm just saying, please voice those, like feel free to add from your end anything that you think would help or hurt your model as it comes together.
Thanks, Krishna.
Okay.
Other updates?
I just had one fun thing to think about. I'm working on a paper and I've been reading a bunch. We've had this discussion of like what is sort of an organism versus an organoid versus a biobot. And going through this, there's a really cool paper that's old, and I'm trying to follow up to see if they followed up on it, but just something to think about. There's a group that in the early 2000s, 2010s, they were looking at cilia beat frequencies and power in mouse airway. So they basically looked at in the trachea and in the lungs of mice, they would open them up and they would measure how fast the cilia beat, how fast they can get them to beat by stimulating them with things to make the airway clear, and how slow they can go. So basically this idea of a range of motor speed. And they got an upper and lower bracket. The same group, what was cool is they said, well, let's take out that lung trachea and grow it in a dish with culture media, basically anthrobots. And they did it flat and they did it as spheroids, which are anthrobots. And so this is a very different system. This is sort of like what Ali's doing. So in the body, right, these are getting blood supply. There's different cell types present. In a culture dish, these are submerged in just liquid energy with some carbon dioxide, and they're basically good to go. And what was cool is when they did this, they actually found that in the dishes, everything ran at maximum speed all the time. There was no modulation. They're basically like running the engine with the pedal to the ground because you've just got unlimited energy. And they really struggled. They couldn't find a way to even slow them down. So I have not looked at this explicitly in xenobots. I think that's different because there's no culture media. These are just skin that's facing salt water or an organoid in salt water, but we don't know. We've never carefully looked at that. But in this design space, just thinking about that might be interesting, right? So that might say that one of the goals should be, rather than worry about speeding up cilia, we should actually focus on slowing them down because we're now at 100. So we have a lower basement rather than worrying about in the body where it's known how to speed them up. So just thinking about this, like we assume the systems behave somewhat similarly, but and they do overall, but these differences are not trivial and they may inform some of the results. And also if we read things in papers, just being careful that we understand the systems don't always transfer one-to-one between different environments, even within the same animal.
Wow, this is super cool, Doug. I would not have expected that. Again, the opposite of robotics, right? It takes everything to
Correct.
actually get things to move and go. This might be, how do you intelligently suppress?
Correct.
Interesting.
And the idea, right, that in mouse, it's known that, and in frog, there are certain signals that will make the cilia beat faster. But for the wet lab people, at least, if we don't see that in a dish, it may not be because of anything specific on our end. It could be the systems behaving differently. We're running the engine at a different baseline.
And actually, that reminded me of a question that I had for Elena, where we're struggling with, bringing the bacteria and bots together safely. Elena, do the bots that you're working with, do they have cilia? Like, are the cilia recognizing the bacteria and beating to try and like get the bacteria off themselves?
Yeah,
Do
it
you know?
depends. It depends on the stage or the bots which I use. Usually for these experiments, I'm trying to use early stage xenobots like day 2, day 3 after making. So they start to grow cilia, but technically not moving yet. Maybe it's beating. I should ask Doug and Pai
Yeah.
if they're beating at this stage. Doug?
So by the end, very, very late in day 2 is when the cilia start growing, but they're not functional. So
Yep.
molecularly, you can detect them with antibodies.
Mhm.
By the end of day 3, the first start beating, but like you said, it's very slow. And then by day 4, you have typical biobot behavior.
Mhm.
That would be on like Sunday at 14 degrees-ish, after you cut caps.
But how about sensing? So if it's not beating well. Is it still sensing bacteria? Or
So
other things?
it's thought, I have to look at these papers, that they can sense and release antimicrobial peptides in the case of pathogenic bacteria. It's unclear for non-pathogenic or resident microbiome how much sensing is going on.
Mhm.
So the reason I mention it is, Elena, at least on our side, we've been brainstorming for your project about how to get the AI to design something in the immediate environment of the bot to minimize contact, to sort of slow the biofilm down. But we could also try and task the AI with helping the bot to fight back. And I mean, Doug, correct me if I'm wrong, but we don't yet have an AI design lever to control the amount of cilia or the force at which the cilia beat. But maybe in the not too distant future we could. So the AI designs a bot that's actively pushing away the bacteria, and I don't know, basically helping it to defend itself.
Mhm.
Is
Yeah.
that something that folks on the Tufts side have thought about at all? Just in general? I don't know if that's possible.
Yeah, so one of the things we wanted to look at, this could dovetail in the MomBot proposal, was looking at if you expose biobots to bacterial compounds, either for long periods or short durations, or pulse it, like, is it like an allergic reaction where they become more sensitive and respond faster or stronger, whatever that may be, producing mucus or bleeding? Or do they attenuate and sort of slow down their response, or neither? Is it just, it's like a machine and every time you dose it, the same thing happens?
Yeah.
That's something we could test out. We have molecular ways to make more and less cilia that are sort of top-down implemented, but I don't think we have a good understanding of how these processes, like, could bacteria stimulate the genesis of new cilia on the surface to help remove them over time.
Got it.
Yeah, and also about that idea with blue light to restrain bacterial growth. I checked our microscope setup. Actually, we have a whole model in our microscope so that we can stimulate with blue light at a certain patterns and also do photo bleaching. Yeah, so it's integrated in our microscope system, so it's doable.
Oh, that's very
Josh.
cool. Yeah,
Yeah.
that's great. Awesome. So just to clue everyone else in, so we've been talking in the Plug and Play Nice project about on the AI side generating CPPNs, and I dropped a YouTube video of a CPPN that paints a blue and black video that we were talking about projecting around a xenobot because blue light tends to inhibit bacterial growth. So awesome, Elena, that you got the equipment to do this. I think this would be a quick win if we can make it work. Very cool. I think it's also it's not within the bounds of the Biosense project, but it is an optical intervention. So I think there's this common thread through everything we're doing. Like what can you do with different modalities when it comes to AI generated interventions?
Elena, have you also tried, so right, there's multiple levels of sensing and or defense, and not that the biofilms are antagonistic, but they're certainly not native bacteria to the frog. So you have the cells that can sense bacteria through surface proteins and release antimicrobial peptides and other things. You have the cilia and the mucus that act as physical barriers to remove pathogens. But it's also thought that the frogs do have a resident microbiome, so their own bacteria, which we know if you swab them and streak this on a plate, they grow all sorts of stuff. They're super dirty. And these are like they're happy to have these on them. I think that it looks like they either come from the egg or from the mother when they're deposited. Have you thought about trying to, at least that one, we could wipe out the resident microbiome? Like, do you think that it could also be that the native bacteria are sort of acting as a second level of insulation?
Yeah, it could be. Yeah, I think we discussed this, that we can remove intrinsic biome in xenobots. I didn't try yet, but yeah.
Cool.
Do you know, how about in the opposite direction? If you put if you take your bacterial media and you put in a biobot with no biofilm, will the local microbiome start growing like crazy? Like, are they essentially competing with one another in the environment that you now are sort of releasing the local microbes that have nothing to do with the biofilm, and they're now competing for growth and reagents? Like, have you ever tried just sticking a biobot in your media and see how fast it grows junk?
I did. Bot dies very quickly in normal bacterial media. Usually for my experiments, I use minimal bacterial media. But if I use LB broth,
Gotcha.
where I grow bacteria and I put Xenobot there, it's just death in a couple of hours. If it's due to what bacteria it's happening, yeah, basically if I don't put bacillus there, it's due to own bacterial growth. But which bacteria involved, I'm not sure if we can analyze that. How to analyze bacterial species?
We have a collaborator here that can do
Yeah.
that, so we could plate them and just try to do visually, see what kind of things are there. But there's also a sequencing facility for bacteria, sequencing facility to look at the microbiome that will give you the different community members. But it's just one thing to think about, because you're doing bacteria and biobot, but there's also the third thing, which is the local bacteria on the biobot that could be affecting the experiment.
Yeah, it's definitely, I think it's a very complex communication when I put Xenobot and my bacillus in a dish. It's communication between all three agents, internal microbiome, frog cells, organoid, and bacillus. And who is killing Xenobot, we don't know. Maybe not bacillus at all, maybe only microbiota is doing that.
Yeah, Mike published a paper like 10 years ago that showed that the bacteria, and there's actually a couple of new papers since then, the bacteria on the surface are actually required for regeneration in the tadpole. So if you cut the tail, the tadpoles regenerate. If you kill the local microbiome and cut the tail, they do not regenerate. So the bacteria, it's like mycorrhizae that help plants grow. These local microbiomes are producing things that we think the tadpoles can't make on its own, like butyrate and these very unstable molecules. And that, it's just one thing to keep in the back of your mind.
Yeah, and in our experimental setting, it can go in an opposite direction technically, because when we put like special media, xenobots are not used to grow in special media with carbon, nitrogens, or supplements. And if we put in this bacterial stuff, then just internal microbiome starts to grow faster, probably can also kill xenobot. Yeah.
Super interesting. Other updates? Okay, if there's time. So I just, actually one thing I wanted to share about the Giving Cells a Voice project, or the Zap GPT project. I know Mike's folks who work on that are not here, but just been rolling this around in my head. I have a postdoc who's arriving in a few days who's going to be working on that project. And this is, this vague idea of mashing up LLMs and bioreactors. Thomas had some fun wrestling with LLMs to try and figure out how this would work. So I've been trying to think this through a little bit. And on the AI side, I was stuck at least on the issue of we want to issue prompts to the biologicals, via a bioreactor or via Mamba. It doesn't really matter, where we need something that translates English into interventions, electrical interventions, chemical, what have you. But in my mind, there were two sets of things the humans could say to the cells. They could either give a command, they could say, generate this information flow among the calcium in the bot, or whatever it is, do this, do that. And then there's also interrogations where you ask the cells a question. Why is, I don't know what, protein XYZ, why is it so low? And it seemed like these were two separate things. These are two separate types of things you can say to the cells that need to be turned into an intervention somehow, and then you get an answer from the cells. But I was thinking that when you ask a question, why is protein XYZ so low? Why is the concentration of this so low? The cells could quote unquote answer you by showing you rather than telling you. The cells, when you ask the question, that question could be turned into an intervention that causes the cells to collectively stop the result that you asked about and then turn it back on again. So if you ask a person, why is this happening? They can show you by doing something that causes the result to stop and then causes it to turn back on again. And ideally, they do it repeatedly. They do it from a bunch of different initial conditions. They basically prove the causal mechanism to you. So I thought that was kind of cool because there's some interesting things on the AI side we could do. And it would also make that project a little bit more streamlined in that everything that someone types in gets turned into an intervention that causes the cells to do something. And it can be, commands, imperatives, do this, or interrogatives. Why is this happening? Anyone have any thoughts on that? Still filling in the details of how we would do this, but that seemed to make more sense to me than what we had before.
On the bio end, when these approaches are taken, so if you say, right, like, let's say a toy example that's more up our alley, like if you ask a bunch of cells, how do you make an arm, right? That's like the kind of question we're asking. How do you get from a bunch of cells to a hand with digits? We usually do the same thing. We apply interventions that turn on and off individual genes, pathways, mechanical forces. And we usually go down this sort of pachinko machine of two levels. First, we say, what is necessary? And that's often a list of many things, right? So if we knock this out, you get no hand. If you get knocked this out, you get a mitten. If you get knocked this out, you get thumbs, 5,000 thumbs.
Yeah.
Okay, so all of these things are necessary, but they don't make a hand. But these combinations together are then what we call sufficient. So you say this group of things is fully necessary and sufficient for X. And I think MomBot can probably, that's sort of the biological version of what you said. It can start applying interventions to determine which subcategories of things or proteins or whatever are really required. The hard part about that is getting the bioreactor, right? Like imaging and probing and having reporters for those things is the hard part biologically. And like Elena was saying, there's a lot of weird feedbacks that can give you false positives. So you're like, oh, if we look for this with this protein, it works. But then maybe it turns out that the blue light's actually just killing the microbiome and it has nothing to do with the protein of interest. So I think that the validation part's going to be the holdup, the slow part to see, is the AI correct? Or is it generating some sort of false positive or negative?
Totally agreed. And I think this is certainly not anything that's going to be done within the scope of this current Zap GPT award, but like the sequel or the one after that is the interventions are like the things that get generated by the human generated question is to do all those things in as many different conditions as possible. So the cells are being tasked, or the AI is figuring out how to task the cells to show you, to rule out, conflated variables and, unobservables and all the rest. Can you do it? Can you grow a hand at night and in the morning? Can you grow a hand at 10 degrees, 20 degrees? Can you, all that sort of thing. that's show and tell. If you really understand what grows a hand, you should be able to do it in as many different conditions as possible. Like fill out the attractor landscape for regrow a hand as fully as possible.
The other thing this might help with conceptual wise is biologists have tried this idea of doing what you're doing, but from the top down, right? Building synthetic circuits of on and off. What's hard is that, as we know now, is gene regulatory networks are usually tens of thousands of genes, and most of these things are very promiscuous. So if I turn off this gene that I think makes a hand, it will turn off the hand, but now you have two heads because you didn't realize it also plays a part in these other pathways. And that might actually be better for the AI. The humans are bad because we're very bad at conceptualizing these really loose regulatory networks that are very promiscuous, but the AI might not care, right? Like it might just say, I can look at 26,000 variables simultaneously and build a principal component analysis from that. So yeah, absolutely. I think that would be exciting. It's just a matter of how many things can we measure, I guess, is the question biologically, because if we can only get you calcium data in one chemical, there's not a lot to ask the machine to do necessarily.
True. And I think I forget the Yanbo, and I forget the other person's name there in the Levin lab who's working on this. But that's the thing, right? You want a simplified environment. Like it would be great to do this with the Lac operon or Lotka-Volterra equations and go from there. But yeah, interrogating a 10,000 plus gene GRN is going to be tricky. One other just miscellaneous item about that project, just wanted to share that with folks, is when the cells do something and we get the times and we get the MP4 back, the video of what the cells are doing, that there are LVMs, large video models now, and this was an idea from one of our colleagues, that they'll annotate or they'll describe what's going on in a video. So if you take a video of the xenobits, it'll say it's a whole bunch of little bits of things congealing together. Like that's the description. You can get it from an LVM. And now if you imagine this pipeline where at the start of the pipeline, somebody said, hey cells, please aggregate, which generates an intervention, and at the end, the LVM generates a description of the video that says there's a bunch of things that are aggregating or coming together. You now have English sentences or short phrases at either ends of the pipeline, and you can take the word embeddings of those. This is the AI, the AI view on words are just vectors, and you can actually take the distance between the vectors, which gives you a loss value. It tells you how well that whole pipeline worked. And in theory, you can then propagate all those errors back into all the way to the most upstream side, which is how the intervention should have been different given the command to aggregate the cells, and then forward and backwards. Yeah.
I was going to say, this reminds me of, is this that same video program where you can feed it the beginning of the video and then let it run and it will tell you what it thinks should happen with like human videos? Have you seen this?
Yeah, I mean, there's a ton of those now, right? So there's a proliferation of them. And so those are ones that are like, they're telling you what's in the video by guessing what's going to happen next. Like it's like Krishna's model, but on steroids.
But I'm thinking that's the similar type of thing, right? That you would need cellular data for this, but this is an ugly example, but it's just one that's been going around. Is they fed it in this famous video of Chris Rock getting struck at the Oscars, and if you feed it the first half, the two people just come up and hug each other, and so it realizes like. But it's interesting because it tells you, right, that there's an unexpected event there. Something happened that did not fit the model. And so I'm wondering, the biological example of that, right, is it builds models and it has expected features and it can say, aha, this is where two cells should have hugged, but instead something weird happened, and feed that back to the scientist.
Yes. Yeah, absolutely. Exactly. In my head for the moment, it's actually video to text, right? Like the thing generates a text description of what it thinks is going on in the video. But yeah, we could do a lot of this, but at least at least there's less work for us on the back end, that we can plug something in that generates plain English text that we can compare to the original text. That was the piece in my head that was missing, is that gives us a loss value.
So Josh, I don't know if you remember this, but what Doug is describing, they're like, oh, I'm making a prediction and like that's where it's wrong.
Yeah.
I don't know if you remember all the negative local mutual information stuff that I was working on with the Lotka-Volterra system.
Yeah.
So that that turns out to be much easier said than done, because it requires doing things like defining a bunch of priors and like figuring out, well, what is it that you believe? So like I still think it's a good idea, but I just wanted to make a note that like I have seen this movie before,
Yes,
and
yes.
it is kind of a horror movie.
Yeah, yeah, absolutely. So the other thing is no LLMs here. So I'm thinking that we just use the old word embeddings, word to vec. So, only one word or two words are allowed. It's not full, sentences and all the rest. We keep it very, very simple.
Oh, that could be much more tractable then.
I think so. I mean, it's still definitely non-trivial. There's still, it's an underdefined space. But I think, yeah, if we can get all this to work with, one word, two word prompts and then responses and get any of that to work, we can let someone else who's more comfortable with LLMs take it from there. Okay, just wanted to share, just bounce that off, folks. Anyone else with updates? Okay, we got 6 minutes left. Can I ask that we use these 6 minutes? Because then Doug and Krishna and I have to go jump to the Boston engineering meeting about developing the interface between Medea, our AI, and Mombot. And we've got as far with Boston engineering as making it clear that Doug, or at least some human in the Levin lab, goes to Mombot, fills up a dish with fresh frog cells, and then presses go. And at that point, the process becomes an automated back and forth between Mombot and Medea, generating and applying interventions back and forth, back and forth. But it's not clear to me where that process ends. There's in Mombot, there's two sets of dishes. There's like the nursery in which aggregation and xenobits come together into bots, and then at some point, bots are dropped into their jungle gym where they go about doing, live out their adult lives. I'm just a little fuzzy on those later stages and thought we could just talk through them for a few minutes.
Sorry, I had to step out of the office because there was construction going on. Can you just repeat again the part
Yep.
that's unclear?
Yep. So Doug, you press a button, you fill up a well with fresh cells. Medea gets the video, generates an intervention, zaps the cells, zaps the cells, and at some point those cells aggregate into a motile bot. When or who makes the decision about when to transfer a bot from a nursery dish into a jungle gym dish?
So right now, theoretically, that will be made by Medea on the fly on some level. So the idea was not bot in my head is non-moving. So a ball of cells is not a bot. It's just a ball of cells.
Okay.
So there should be almost zero XY displacement of the bot between any amount of imaging that we're doing. Once we see positional, I think positional velocity or positional, Krishna had a term for that I liked. Once we see some positional velocity above some baseline rate, then the bot can be marked as ready for experimentation, and a machine will come like a claw machine in a game and go down and pick up that bot and move it to an arena that has whatever we want in there. And then we can specify if it stays in there forever or if it is observed for a set amount of time and then moved back to its incubator. But I believe that the presence of movement is what we are using to define as being ready for an arena for experimentation.
Got it. Got it. Okay. Okay.
And we only did that because one of the control knobs that we have is going to be a huge range of temperatures. So from refrigerator up to sunny 85, 90-degree day. And that will dramatically change the rate that it takes a bot to get to movement. And that, so for example, it will be 3 to 4 times faster at high temperatures. So it may only take a day and a half at 24 degrees before we have moving bots. Compared to 10 degrees, may take 11 days before they start moving.
Okay, that's super helpful. That's super helpful. So if Medea is responsible for detecting movement and indicating it's now ready to go, presumably Medea can also then say, well, this bot is no longer moving, kill it off, clean the dish, and load it back up again.
Correct.
Okay.
Unless we want to have control experiments where we apply interventions to non-moving bots.
Also true.
Yeah, okay.
But Medea will be making those decisions ultimately.
Okay, I think that's great because then that keeps you, Doug, out of the loop until, I don't know, right at the end. You have to go in and scrape the cells out or not?
Yes, and at least what's good on the beginning is if we start at the temperatures we use in the lab, we can see if Medea is doing it correctly, because I know when the bots will start moving, I know when they will stop moving, and as long as we are within some small standard deviation of what that is, then we're good to go. If Medea is saying after 3 seconds in the dish, I'm ready to start an experiment, we know that something's wrong with whatever's happening.
Got it. Okay. All right, Doug and Christian and I have to jump. Thanks, everyone. Fascinating as always.